Welcome to the Cannabis Equipment News Podcast.
Hi, I'm David Manti, and welcome to a new episode of the Cannabis Equipment News Podcast. With me this week is Kevin McKernan, CSO and founder of Medicinal Genomics. Thank you very much for joining me today, Kevin.
Thanks for having me, David. I'm excited to talk to you about this.
I'm excited as well, but before we get started, please make sure to like, share, and subscribe to the podcast. You could also help us out a lot by leaving the podcast a positive review on whatever platform you use. If you want to email the podcast, you can reach me at david@cannabisequipmentnews.com with email the podcast in the subject line. You can also subscribe to our daily newsletter. Make sure you get it delivered to your inbox first. Kevin, I always like to start with a little bit of background. How'd you get your start in cannabis?
Oh, that was back in 2011. So I had a few friends who were struggling with cancer and had forwarded me a Guzman's paper showing that cannabinoids were anti-neoplastic, which I was completely unaware of. Started digging into that and decided that we had to sequence the genome of this plant because it had not been sequenced at the time. And that could perhaps help us breed for all these extinct cannabinoids that we believe were bred out of the plant as prohibition pushed everyone to breeding for mostly THC. And so, as many people in the space know, THC is often competing for a precursor known as cannabigerol, and some even upstream of that. And so, when you breed for exclusively THC, you usually lose CBD and CBG and a variety of the other cannabinoids from the gene pool. Those have thankfully been bred back in recent years, as people can see. And so getting the genetic pathway of that in the plant understood really helps breeding efforts to that end.
So back in 2011, where was the plant in terms of what was bred out of it? What was its current state? And, how did you work to sort of revive it?
Well, I can't take a single-handed credit for any of that breeding effort. A lot of that happened despite genomics being in place. People figured out how to breed these things with just looking through HPLC traces. But at the time, it was unsequenced. No one knew how many genes there were involved in the cannabinoid synthesis pathway. We now know there's at least 30 of these cannabinoid synthesis genes. And it was a stage where it was mocked. It was really still back then fairly illegal to perform any of that science. We actually had to start a company in the Netherlands to get some of this work done. And as things liberalized here in the States, we moved those efforts back to the States and continued working on it here in Beverly, Massachusetts. But it was a stage. I can remember when we published the genome online, it was mocked by Nature magazine as being a Cheech and Chong project. And I think only like 5 years later did Nature have a cover episode supporting all the rich benefits of cannabis research. So it took about 5 years for it to flip, but we're now at a stage where I think the scientific community takes this much more seriously than it did in 2011. And there's a rich pipeline of people trying to extract different rare cannabinoids for unique disease conditions. So it's moved quite a bit in 13 years.
Did you expect that reaction, though, at the time, given the climate?
Yes, I did, actually. In fact, when we started doing this work, we consulted with some of our friends in the media industry just to ask, like, what do we do about this? We're bound to get mocked when this comes out. And they kind of helped us craft a message that was really focused on the medical side of this. But as anyone knows in the space, the difference between medical and recreational use is really a blurred line. Many people would argue that recreational use will prevent medical use. You really need some of these in your system. At least some people do, for medical conditions not to arrive. So this dichotomy we have of medical use versus recreational use, I think, is very puritanical and is not anchored in any preventative medicine. So I think recreational use is getting a lot more mainstream. At least you're seeing the uptake in cannabinoid use is mostly in the older generation. It's not really the youth that are jumping onto this. If you look at states post-legalization, youth rates are down, while age 50 and up use rates are up. So the baby boomers are seeing this. They see it. It's a better trade-off than alcohol, and it's a safer alternative than alcohol, and it helps them sleep and helps with a lot of the conditions that they have as people age.
Did you expect that as well? I know that I've talked to a number of people that have talked about how the medical side of the industry has kind of definitely taken a back seat just because of the money in the recreational side of the business, are we losing out in terms of advancements in science and research on the medical side because there's sort of the interest is in the big business of recreational?
Well, it's certainly been eclipsed monetarily. There's a larger market in the recreational side, but and the medical side is facing some other unique challenges. The FDA system we have in place today doesn't really favor these types of botanical drug substances, if you will. I mean, GW's had success there. There's no doubt about that. But that was a very long and hard slog for them because the FDA likes to isolate single compounds that are targeted against a single disease. And we know this plant produces a mixture of different cannabinoids that often need to be used in conjunction for them to have therapeutic effects. So it's hard to get it through the FDA, and it's hard to get it funded to get through the FDA because you are funding a drug that's moving into a market that has a dispensary system that will always be competing with you. So there hasn't been a tremendous pharmaceutical push despite GW's effort. Very little of that has been replicated. There have been some places. I think Tecamolum has pushed some work in the autism space. And there's a couple other companies working to that end, but they usually need to have a very unique cannabinoid blend or something that's changed. They've changed the molecule somehow so that it's patentable and they can survive that long, gruesome process through FDA review. So it has been harder on the medical front. That doesn't mean there aren't medical uses, and many of the medical uses will probably be addressed through the dispensary system long before there's any FDA-approved version of this. So yeah, it's not something I entirely forecasted or could see. It's just the nature of how this market's evolved is that the recreational use took off, and there's a lot of people that are curbing their alcohol consumption based on trade-offs with safer drugs.
So how did you find yourself founding Medicinal Genomics?
It's a good question. We didn't really know what the business model was going to be when we started this. We just knew it was an important topic that was unexplored. And so we sequenced the genome and put it public just to see if it would help buoy the field and see if other markets emerged from that. And one of the primary markets that emerged was many physicians were interested in contamination profiling of the plants and how to grow plants that were clean of yeast and mold in particular. So the number one failure rate for cannabis in testing labs is yeast and mold. It's probably 10 to 15% failure rates depending on the state, and that is an enormous economic impact if you can't grow your way out of this. There have been great papers that have come out recently out of Zamir Punja's lab that show that it is very much a genetic trait in the plant that drives how much yeast and mold a plant has. So you can, in fact, grow your way out of this. You can breed to have plants that have lower microbial loads, and therefore you get a higher yield going through the testing process. So that's an area we didn't anticipate, but we worked on that pretty much out of the gate. Once we realized that was the number one concern we saw from physicians was contamination of the product, we started building PCR tools that could detect this and help labs, quickly assess the microbial burden on plants.
So how can cultivators grow their way out of this?
Well, I would point people to Zamir's paper. He went and profiled, I think in his paper, it was 6 different cultivars that they had been growing in production. And there was, over a 10-fold difference in the numbers of yeast and mold counts that those plants would predictably produce. So it was very difficult to take a plant that had a high microbial burden and treat your way out of it. You could certainly irradiate it and clean it up after the fact, but we know that irradiation doesn't totally remove all threats. It removes the viability of the organisms, but it doesn't remove the mycotoxins or the endotoxins that those organisms produce. So irradiation is a way of mopping up a dirty process, but it's not going to actually strike the root and fix the problem out of the gate. I think what Zamir's doing, which is breeding plants that have low burdens, is a longer term solution to this, because you don't want to have to be irradiating your way out of this.
At a base level, what is genomics science?
It's basically being able to read the code of life and also the programs it's running. So with genomics, you can not only read the DNA of the plant, you can also read the RNA to see what genes are getting expressed at, let's say, after hop latent viroid infection. We've done some work in that area. You can see which genes get turned on or off when you get an infection. That helps you understand the pathway that the viroid is working on in the plant, and perhaps breeding your way toward plants that are more tolerant of the viroid versus ones that aren't. There's also people in Canada doing work. David Jolly does some really interesting work in this field, looking at the genetic loci that help predict microbial burden, if you will. They're looking at genes like the MLO regions and the powdery mildew resistance genes that have been identified. So these are aspects of the genome of the plant that can inform whether this plant's going to be more or less susceptible to particular pathogens. It's very early. We can't perfectly predict which plants are going to be clean of mold yet from the genetics. We're still working on that by sequencing all of the plants that Zamir has worked on, and other plants in the industry that are known to be resistant or tolerant to these infections. But this is the bedrock we have to build. We have to sequence enough of these plants that are resistant and enough of the ones that aren't to build a catalog of the genetics that predict whether you're going to have a high or low microbial burden.
Are cultivators resistant to this type of science, or is the community more or less welcoming of new information to try and move the industry forward?
So I think 5 years ago, the science in the genomic space really could only tell you a few things about the plant. And so there wasn't enough of a reward for the growers to get involved. So we could predict whether you're going to be a THC producing plant or a CBD producing plant from the genetics alone. And we could tell you the provenance of the plant. Like, was it truly an OG Kush? Was it truly a sour diesel? Was it truly a Blue Dream? Those types of questions we can answer. But some of those answers the growers already knew. They knew the background profile of their genetics. They knew if it was going to produce THC or CBD based on their HPLC data. So it didn't offer them a whole lot of benefit to get involved. And there was some risk that people would get your genetics, and maybe those genetics could be used to prosper another grow more than yours. So fast forward to today, there's more and more information we can extract out of the plant that can give you information about like the autoflower capability of the plant, whether it's male, female, whether it's there's some early data now looking at whether it can produce varins or not, these different cannabinoids. Genetically we can determine that, and then all of these microbial susceptibility loci, if you will, or they call them MLOs, which is that's one class of genes that are involved in susceptibility to microbial infection. There's another set that are chitinases and thaumatin-like proteins that actually help the plants fight against these pathogens. So a collection of these genes are now known that are involved in pathogen response. And while that's earlier, that's giving people a little bit more confidence that the information can give them additional insight into how to breed. How do we breed for plants that are hop latent viroid tolerant is one thing that we're working on here. How do we breed for plants that have lower total use and mold burden? Those are questions now the data is beginning to answer.
So is it the strain authenticity that gets people in the door, and then sort of the science behind making the plants more resilient against mold and other pests? Is that kind of where they see the true value?
I think so. The authenticity has been important. We've been involved in one court case where some clones were taken from a grow and were remarketed under a different name, and those growers were able to get access to those materials, sequence them, and compare them to the original, and prove in court that they were taken, that they were identical plants. And there is some intellectual property in this space with plant patents that can protect against this. Plant patents only protect against clonally propagated plants. There's other patents out there that are utility patents that can protect against plants with certain traits, if you will. They're much broader than clones. And so there are some growers that are pursuing both of those intellectual property approaches with at least THC producing cannabis. And the hemp side, there's an additional level of protection with the USDA and there's a seed program they have there, seed bank, where you have to submit seeds. You can't do that with THC seeds at the moment, but you can do it with CBD based seeds. So there's three levels of protection there that people, the Plant Variety Protection Act is the one with the USDA. So there are three avenues that people can pursue. We have actually used our data in court cases. I think one feature of what we're doing that helps prove provenance is that when we sequence someone's strain, we take a fingerprint of that sequencing data and we submit it into a Bitcoin transaction so that it's basically immortalized in a public ledger that nobody can change. And so that gives you a good timestamp on your genetics. Sorry about that. My lights just went out here. A timestamp on your genetics as to when it was bred. And that can be used in court where notaries aren't necessarily friendly to notarizing. The notary system in the States that can timestamp things is a federally run banking system. So you can't really bring a cannabis plant in there and get it notarized, but you can sequence it, and you can etch that data into distributed ledgers that can prove the timing of when your sequence existed. So that data came to use in this particular court case, and the judge ruled that it was sufficient for them to determine provenance of the strains.
So it's not that you're kind of circumventing the patent process, but because it's unavailable, is that what forced you to use Bitcoin transactions?
It was, and you can still use the patent system. You can still use. It's just more expensive than sequencing. All right, so to file plant patents may cost a couple $1,000s. Utility patents can cost $20,000, and the USDA Plant Variety Protection Act requires you performing a dust test showing that the seeds are unique and distinguishable, and they carry a certain trait across many different tested seeds. So there's a program in place that takes time, and still a little bit experimental with cannabis. So we looked for something where you can just perform $500 worth of sequencing and get a timestamp to know your genetics, and that can be done without any third party. So that was kind of the reason for getting it done in the past, is that notarization wasn't going to happen anytime soon, and so we wanted something that could log these things. We of course point people to patent attorneys if they're interested in going that direction, because they can do this with far more teeth than we can. If you actually get a plant patent on there, that is something that is very defensible in court, and many of those have been filed for and awarded. So there's precedence there that the USPTO is accepting of plant patents in the cannabis space.
How did you come up with the idea of putting it in a Bitcoin transaction?
Oh, I'm just a bit of a Bitcoin nerd, I guess. It's a technology that I find is I've always been attracted to it because it looks like a perfect system for peer review. If you can do peer-to-peer peer review without a journal involved, you can effectively get a lot of the attack surfaces on journalism removed. I mean, it may not be as apparent to people in the cannabis space, but it certainly became apparent to more people during COVID. There was just a lot of papers that got retracted in COVID for political purposes. And so the journals are in fact funded by the pharmaceutical industry. Their advertisement revenue is all based on this. And the cannabis plant hasn't necessarily been a friend to the pharmaceutical industry. It has to some people in the industry who've done very well with it in GW, but in the past, it's often been something that competes with other pharmaceutical products. And so, and since it has a generic dispensary distribution system, it's not necessarily something the pharmaceutical companies are gonna buoy up. In fact, what we saw during COVID is many of the generic drugs got attacked, like ivermectin and hydroxychloroquine. And this is just the same story with cannabis, except the cannabis field was aware of it for 30 years longer, that they saw cannabinoids kind of getting shorthanded in the literature because there was no owner of them. They're more of an open source set of molecules that people can grow in their backyard, and no one has strong rights to them. So the pharma industry usually annihilates those types of competitors by putting pressure on journals to have slanted articles that come through. So the Bitcoin system looks like a public ledger that if you can publish proof of your experiments on that system, you can't publish all the data. It's not meant for that. It's not a database that can hold large volumes of scientific data. It can just hold a fingerprint of your data to prove it existed at a certain time, and usually have to host that data elsewhere. But when we saw that one of the cannabis genomes we sequenced was Jamaican lion. It was funded by a cryptocurrency known as Dash, and it was all those experiments were hashed onto blockchains. The project was funded by cryptocurrency. It was recorded by cryptocurrency, and the peer review was performed in a distributed manner where we just put out bounties for 3 reviewers to put up their thoughts on the paper, and all those were etched into a ledger that no one can change. And since then, that reference genome has been used and cited in many other peer-reviewed publications. So it is a very fast and efficient way of getting work done, where international boundaries are not necessarily friction points for moving money around.
Are you drawn to the idea of little cannabis versus big pharma?
Well, I don't call it little anymore, So
yeah, I guess comparatively.
Yes. I am, and that the thing about science is that it should favor non-narrative discussion points, if you will. Discovering new data oftentimes is counter narrative, but what really happens in science is an appeal to consensus of the prior narrative. It's almost like a Gutenberg era where the large incumbent companies get to dictate what the narrative is, and cannabis is counter narrative to all of these discussions, and so it's very hard to get a message out in the cannabis field without the incumbents pushing back on it. And science to me is all about new narratives, is about challenging the narrative. About if you can't question it, it's not science. And so, in many ways, I think cannabinoids offer this hope to a lot of patients who see the FDA as a stranglehold on their access to the right to try new medicines. So I'm very much in favor of personalized medicine and people being able to control their destiny and pick their own therapy, if you will, regardless of what the FDA has to say about it. And so cannabis fits that bill quite a bit, as does a lot of things that we saw come through the COVID debacle, is that there's a lot of drugs out there that could have prevented this mess, and they were systematically attacked and destroyed just like cannabis was. And that's something that is a violation of everyone's sort of sovereignty and their medical freedom, if you will.
How would you describe the state of cannabis science?
Today it's growing quite a bit. It's an international effort, and we are seeing more and more of this get through the peer review process, which is fantastic to see. So it's certainly changing and becoming more accepted, if you will, inside of what we would deem the narrative. But I think it still has a long way to go. There's still obviously hindrances on doing any type of clinical trial. We're still in this world where the DEA has a stranglehold on what cannabis can be used in a study. Most of it's pointing back to Mississippi, which has very low cannabinoid content cannabis that's not really reflective of what's in the marketplace. The total use and mold levels are fairly high from that material. So we really want to be able to see studies done on cannabis that people are using. I mean, there's a whole debacle in the cannabis science community over right now over sort of schizophrenia induction. And a lot of that, if you look through the literature, it's funded by NIDA, who has a charter to look for drug abuse, not drug benefits. And it's often confounded with a lot of genetic susceptibility, if you will. A lot of schizophrenia has a genetic component to it. And what people are finding is that cannabis doesn't necessarily induce schizophrenia. It's that people who are genetically prone to get schizophrenia tend to have drug-seeking behavior, and cannabis is one thing that has literature suggesting CBD may actually reduce psychosis. So a lot of these schizophrenia papers, they look at cannabis, in fact, marijuana. They don't even call it the correct name. And they never chemotype the samples. They never pesticide test the samples. They never look for the total use and mold in the samples. And all of these other attributes in the marijuana have literature ties to some pesticides causing psychosis. We got some microbes that make mycotoxins that can create psychosis. So we don't have a clean picture on this association. And likewise, we tend to measure one side of the equation through many NIDA-funded grants, which is let's look at the number of people who were not exhibiting schizophrenic-like characteristics, and then they start using cannabis and get them. What they don't find in any of those studies is the people who were schizophrenic prone and used cannabinoids and never got it. That doesn't show up in their data, right? And if you look at the schizophrenic rates between countries that are neighboring one another with similar societal things, like you can in like Scandinavia, you'll see that the schizophrenia rates haven't changed despite cannabis use going up dramatically in neighboring countries. And so that tells you that there's no net change in schizophrenia with cannabis use. So maybe some people are prone to it triggering it or being involved in their genetics. And there's an equal number of people who are benefiting from it that are going unrecorded. So it's a complicated field, particularly if you do any epidemiological studies on this, because there's a lot of confounders in this, from socioeconomic status to background genetics. But it's an important part of the scientific literature, because we're seeing this get thrown at us as cannabis gets more popular. We're getting people like Alex Berenson and others coming out saying it causes psychosis and violence. And a lot of scientists who he references claiming that he's completely misinterpreted all of their work, where their work was actually proving the opposite of what he's claiming, which is that it has a genetic predisposition, and cannabis is just one of the many drugs they may choose to use in their seek for something that alleviates their symptoms. So that light is on an auto thing. Sorry, we're very photon
No, no
cautious.
worries.
It's going to go off a couple more times if I don't keep moving in very animated fashions here. So anyway, but back to your question is that yes, it's improving. The science is moving in the right direction, but we have to constantly be aware of the bias that is in the scientific process, which means it tends to be industry funded. The journals have influence based on a particular narrative and by the people who fund them. And oftentimes we're not measuring for the benefits. We have full government agencies that are only looking for abuse. And so we end up with things like cannabis use disorder, right? Which is one of these things that they don't apply to food. They don't apply to Adderall per se. I mean, there's abuse potential there, but they have kids using Adderall in school, and you don't have this. If you use Adderall every day, you're considered an Adderall use disorder, but you do have that on cannabis. There's a cannabis use disorder. If you use it every day, you're considered a cannabis use disorder. That is a phrase that's really being born out of a government's agency's interest in proving abuse. And it's not grounded in looking at this from a holistic picture of benefit versus abuse.
So you think that this use disorder could be pretty much found in anything from food, Adderall, or any substance, and it's just that certain agencies are making sure they focus on it with cannabis?
Yes, there's the motive out there at least from the literature that I scour through. They tend to have grants from NIDA or from other government institutions that are trying to point out the abuse potential that's going to occur with legalization. And it's very rare that the same papers will stop and look at the benefit side of the equation, that, okay, alcohol use went down in these jurisdictions. And so this cannabis use disorder nomenclature is a really large catch-all that if you applied it to other drugs, you'd be horrified. Everyone would have a use disorder for all the other things that they're taking. So I don't think it's a very productive approach to vilify patients like this, because many patients may have an endocannabinoid deficiency and they actually need to take some of these every day. It doesn't make them it doesn't mean they have a use disorder. It means that they're balancing their endocannabinoid system back to baseline, and it happens to be therapeutic for them. So it has a little bit of a puritanical touch to it, I think.
Do you find yourself more encouraged by new studies that are published, or upset by studies that you poke holes into?
It's every study can have holes poked into them. I think the important thing to read in any study is first read the conflicts of interest section. You have to find out who funded it, and then the abstracts and conclusions are important, but really the methods are what matter, because the abstracting conclusions are often crafted by the editors to meet narrative, and oftentimes the methods don't support what are put in those paragraphs. If you read through the methods, you can see they can't really be making those claims based on what was presented. They're just being pushed into fitting a popular narrative, if you will. So it's important to dissect papers very closely in that regard. But I'm more the papers that I gravitate more toward ones that are looking at how to grow for more effective yield, and the things that are sleuthing out the different genes in the plant that might be performing or be giving us some guidance on how to breed these things in directed fashions, because those typically don't have any of these socio-political bent to them, if you will. They're just purely looking after what gene causes X and what gene can predict outcomes of yield. And they're usually not tainted with, they're not designed to make headlines that will get waved around the New York Times about how all the kids are soon going to be, consumed with new diseases based on cannabis use. They're simply looking at really the structural components of what we can learn from the plant.
When it comes to learning from the plant, what could cultivators do that's sort of the low-hanging fruit when it comes to improving yield, safety, and quality?
Well, yield is an important aspect given the financial circumstance in the field. We have a banking issue in our field where it's very difficult to get funding. We have a tax issue where you can't write off your business expenses, and that might change. At least the 280E aspect might change with the descheduling, which would be very helpful for the field, because I think the field will then have a lot more money for research. Right now, I think a lot of people are just scraping by based on the financial screws that are attached to the industry right now. So I think that's an exciting part of it. So whenever you have these tight financial constraints in the field, yield becomes incredibly important. How do you grow things that are clean? without resorting to lab cheating, because there's a tremendous amount of that going on in the industry right now. You can actually look throughout a lot of the metric data that's come out of various states and see that there are some labs out there that never fail samples, and that's just not possible. They're clearly, and they're gobbling up most of the lab testing business, because these grows are having to compete with one another. And if they can go to a lab that gives them a 2% failure rate versus one that gives them 10% failure rate, all the business gravitates to the lab that cheats. So that's an area that we have to address in the field. Otherwise, the testing won't be meaningful at all. And if that's the case, there's not much of a differentiation between the black market. Last numbers I've seen is the black market is still the majority of the cannabis being sold. So despite legalization, we're making a dent in the black market, but it's not denting it fast enough, probably because the taxes are very high, and it encourages people to work outside of the system. But as those taxes lower in time, I think more and more will move to the regulated markets, where the main thing that differentiates, I think, the products are, is it clean or was it loaded with pesticides and mold? And do you have an accurate label on it in terms of THC and everything else that's present? Right now, we have an industry that I think that's caught between this black market pressure and trying to appease growers who are working the regulated system, where the growers really need high yield, and that comes down to these tests, unfortunately, whether they're accurate or not. That's the largest reason for failure in the field are the microbial tests. So they have to find labs that have really low failure rates, or if they're honest grows, they will figure out how to grow cleanly from the start. And that takes more investment and more time. And sometimes it requires some sequencing to really understand whether the genetics of your plant are up for the task. But if you can do that, that's the better manufacturing approach, because you're not mopping up a mess afterwards with irradiation or lab gimmicks. You're actually setting up an operation to grow cleanly from the start. And once you do that, you won't have problems going forward, and you can get predictable yields. So we think the longer term play here is breed for plants that have low failure rates, and you won't have to get yourself caught in this trap of running around to labs that pass and then having there are regulatory agencies now cracking down because they're finding all this cheating going on, and eventually that's going to smear someone's brand. They're going to find a lab that's been cheating, and they're going to go to the growers who are constantly going to those and start knocking on their door saying, hey, how come you gravitated away from labs that were giving you honest numbers to ones that were always giving you passes? And it's starting to unravel right now. There's a lot going on in California on this. There's a lot going on in Massachusetts, Michigan. Many states are seeing that this is happening, and the regulators are starting to react.
I understand that they can help protect themselves on the genetic side early on in the process, but how do you feel about later in the process adding something like a kill step?
Well, I'm not adverse to kill steps because it does destroy the viability of the microbes, and that's a good thing. But it's also a way that you can hide a lot of risk. So if you kill the microbes, that's we don't want them around. But if you get used to that and you start growing stuff that has 1 million CFUs and you irradiate it into non-detection limits, it still has the mycotoxins and the endotoxins that people aren't really thoroughly testing for on the plant. And that can pose risk. So this isn't theoretical. A lot of people in Oregon are upset about this because they had some microbial limits put on them that tightened up the regs in Oregon, and they sued the regulators, and the regulators folded. So there's very little microbial testing in Oregon right now. Endotoxins is something that we have not, as an industry, really paid attention to, and I think we should, because this is something that's going to show its head in this world where people irradiate everything. Because the irradiation doesn't kill the endotoxin; it kills the microbes and our ability to detect that it might be there. But there are endotoxin tests out there. We've run a few of them. We've seen that the endotoxin levels on a lot of cannabis is really high, and endotoxin is known to trigger asthmatic reactions in people. So the CDC also did another study where they looked at the insurance databases, the ICD-10 codes and insurance databases for cannabis users versus non-cannabis users. And they found there was like a 3-fold higher rate of aspergillosis and mucormycosis and other fungal infections in cannabis users. So there is something happening at an epidemiological scale that they can detect in the insurance databases with cannabis users. There's more fungal infections going on. So we have to really start paying attention to these microbial loads and whether we want lab shopping to occur, so that people can find labs that have low pass rates on this stuff. That's going to allow a lot of the stuff into the marketplace and probably contribute to those numbers the CDC is seeing. And let's just hope it doesn't create another asthmatic attack from other patients or other employees in different grow environments. We did put out a white paper on this. I'll forward people to. It does look at endotoxin levels in cannabis, and it also points to all the other studies that looked at this. There's a couple other OSHA studies that looked at endotoxin levels in trimming environments in hemp grows, which are outdoor grows. But the trimming environments there, they had like 40% of the employees were exposed to higher than the DCOS limits, which is like 90 EUs per cubic meter. And so there is an exposure of endotoxin. It's mostly coming from gram-negative bacteria on the plant. And there are biocontrol agents out there that are gram-positive, like Bacillus, that push out these gram-negatives and can probably lower that burden quite a bit. So there are grow tools out there that can help suppress the bad bacteria, if you will, and replace it with healthier bacteria for people at least. And I think that's an important area that the field can leverage with genetic sequencing. Well, one thing that wasn't evident to people sequencing with us 5 years ago is when we sequence, now we mostly do whole genome sequencing on cannabis plants. And when you do that, you get a picture of the whole microbiome with it. So we can tell you how much Pseudomonas is there, how much gram-negative bacteria is there, how much gram-positive material is there. We can tell you how much yeast and mold is there. And that helps growers tremendously in looking at what they're doing in their process. If they want to get through these yeast and mold tests, which bacteria and which fungal microbiomes they can tolerate in the process of growing for yield. But I think your question came back to yield, and yield is very key in a competitive market like this. And there's obviously yield and flower mass. That's a whole nother side of genomics that's still being unfolded here. But something that's a little bit more evident today is we can catalog all the microbes that are on your plant pretty readily with sequencing.
Please send me that white paper, and I'll make sure that there's a link in the description so everyone can find it really easily. Can you describe, so what are endotoxins and how do they proliferate on the plant?
So it's gram-negative bacteria have these in their coat, and it's a lipopolysaccharide, and it induces a cytokine storm if it gets into patients. So you get a very strong immune response. Presumably, when they've seen this actually the tobacco industry had a problem with this, and a lot of endotoxin comes through tobacco smoke. So it's not something that gets killed with heat. In fact, it's autoclave resistant. So I would imagine it's burned and it gets destroyed at the tip of a joint, but the rest of the joint is getting just smoke, aerosol through it. It's probably liberating some endotoxin. So we're setting up to do some experiments right now to look at how much of the endotoxin is making it into the smoke stream. Right now, the only data we have is how much is on like a gram of cannabis. We've taken samples here that have gone through the regulatory environment here where it's tested, and stuff that's come from the hemp market where there isn't any microbial testing. And they'll have very vastly different CFUs, which is viable colony units on plates. The stuff through the regulated market will almost be clean of it, like it's been irradiated, and the stuff in the non-regulated market will have really high levels of bacteria. If you go and look at the endotoxins, they're the same. All right, we get the same amount of endotoxin coming through the regulated market as we have in the unregulated market. So the processes we have in place to kill the microbes aren't getting rid of the endotoxin. How much of this is tolerable? Well, we don't know yet. I mean, cannabis use has been, it's been throughout time and quite prolific, and we don't have a lot of cases in the literature of asthmatic attacks after use of cannabis. So clearly this is not hitting everybody. But there may be a set of people that already are prone to this, that already have asthma, and this is just one more hair on the camel's back that triggers their attack. So for anyone who has asthma, they should probably be very concerned or aware of these whoever's decontaminating stuff, because the decontamination process is going to hide this risk. In fact, we would argue the right thing for the field to do is to put the CFUs before irradiation on the label and after. So people know, all right, was this a 100,000 CFU sample that I got irradiated down to 1? Or was this a 10 million CFU sample that got irradiated down to 1? So you can know what the prior burden was before the irradiation occurred. And that would be the most transparent thing for the user, so they can decide whether they want to touch any samples that were heavily contaminated.
I've been asking this question a lot, and it's just odd how persistent it's been, but are you surprised by how much the industry fights against growing cleaner cannabis, or selling remediated cannabis, and further like truth in testing?
Yes, I mean, I understand it from an economic standpoint. In a scenario where you don't have banking and you don't have tax benefits like 280E, everyone's competing financially, and so they get painted into corners where they have to cut certain corners to survive. So maybe it's not that surprising. We should, I think, turn that back onto the regulators to say, hey, let's not create environments that do this. All right. One, let's get rid of this 280E thing as a mess. That's really making it difficult for businesses to survive. And if you want the black market to go away, you can't be having, tying up one hand behind the legal growers back and asking them to survive. So they're very much fighting economic pressures from the black market. They're trying to do the right thing, and yet the regulators aren't necessarily policing the black market out of existence, and they're not policing the labs that they are being forced to test through. So as a result, the growers just gravitate to the labs that pass the most material and give them the highest THC levels. The interesting thing that we can see in the data from the states that we've collected is that there isn't really any evidence of people lab shopping off of the low failure tests. So E. coli and Salmonella don't fail very often. They're just not found frequently in cannabis. No one lab shops off of those microbes. Total yeast and mold, there's clear evidence of some lab shopping going on. Aspergillus depends on the state and what rules they have in the state for testing Aspergillus, but there's some evidence of Aspergillus shopping going on because the fail rates tend to be in like the 2 to 4% range, and sometimes that's enough for a grower to try and find a lab that never fails. So it's very dependent on the microbe that's being tested. The low frequency fails just don't hit the radar hard enough for anyone to care about them. It's really, we see total yeast and mold and some of the other total tests. Now there's a strong argument. I'm in the microbiological space, right? So I don't actually think the total yeast and mold and total aerobic tests are very helpful, even though they're in regulations and we make tests for them because we have to. They're not helpful because they don't tell you, they don't itemize pathogenic versus non-pathogenic organisms. And if you want to use like biocontrol agents like Bacillus, which is gram-positive, you'll get penalized for doing it because it will elevate your TAC counts, depending on your jurisdiction. Some jurisdictions don't have that test, but so these aren't very informative tests from a clinical perspective. In that, if you read clinical literature, it will always have a Cox postulates in there, which is that we identified and isolated this organism in this patient and it killed them. So that will be written in the language of like Pseudomonas aeruginosus killed this cystic fibrosis patient, right? Or Aspergillus flavus killed this particular, there's a Remington paper that showed one of the Aspergillus species was in their vaporizer from cannabis and it killed a patient. So that's how all the clinical literature is written. And if you want to be able to defend tests long term, because growers do sue regulators when they think they have these things wrong, you have to have this clinical literature to back up the fact that there's a pathogen in here that is known, identified, isolated, can satisfy Koch's postulates, and therefore is the clinical risk. When you get involved with these tests like total aerobic counts and total yeast and mold tests, you're never identifying anything. It's really a tarot card of microbial burden, and it doesn't even measure totals. It claims to be a total test, but only 5% of the microbial species can be cultured. So you really have this partial yeast and mold test and partial aerobic count test, and it doesn't really itemize anything. So you'll never find clinical literature showing that this patient died from a 10,000 CFU total yeast and mold plant, right? It's just not specific enough. So they're not very helpful tests. We've always been trying to encourage regulators like define the species so it will hold up in court, and that both sides of this equation can be satisfied, right? The growers can be satisfied that you've identified a species as pathogenic and you can detect it, and the patients will be satisfied that there's clinical literature that can guide them. When you get involved with these total tests, none of the clinicians understand what it means. I know of many clinicians that have gotten into the testing space, and they're like, these tests we laugh at in the hospital because they don't identify the pathogen that we're talking about. They just count total amounts of bacteria and fungi, and we're not even itemizing which ones are good or bad. So I think long term those tests are actually bad for the industry because they are not going to allow labs to use the greenest technology out there, which is to use Mother Nature, against Mother Nature, if you will. Use beneficial microbes to take out the pathogenic ones. This is what you see happening in big ag is to get rid of Aspergillus, they don't fight it with chemicals, they fight it with AF36, which is an Aspergillus flavus that doesn't have the toxin genes and outcompetes the ones that do make the toxins. And so they can apply those beneficial microbes, there we go again, and not get penalized. in any of these testing regimes. So right now we've got a scenario where these total tests really limit a grower's capacity to use biologicals, which are probably the greenest way to do this. We don't want to be encouraging people to be going down the route of using some bizarre fungicide that's not on the list of things that are being tested, which is what can happen if you put up a list of 66 pesticides you can't use. Sometimes growers will say, fine, I'm going to use one that's not on the list, and then no lab's going to see it. And that's pushing them into a kind of a cat and mouse game that will never end. Just like with synthetic cannabinoids, they keep changing one carbon group on those things, and they're not on the list. So I think we want testing in place that will enable the field to use biological control organisms in the future. And right now, the testing we have that doesn't really differentiate these things, I think, hinders that. So we're more advocates for really species-specific testing, identify the risks that are on cannabis, and then target them with tests that presumably will have a very low fail rate because they're only going after single organisms, but they'll be very specific in known risks that are in the clinical literature.
Would it be simple enough to usher in this species-specific testing that labs could adopt it easily, or would it be a pretty brutal undertaking?
No, actually, it would be a hell of a lot easier, mainly because labs can agree when you define what species you're targeting, you get much higher concordance lab to lab. You'll see all of the lab debates or lab fights, if you will, are around the total test, because depending on the technology you use, you get wildly different answers. And that's mainly because if you grow on one media, you'll get a completely different picture of the microbial content than if you grow on another one. So some states are trying to anchor their total yeast and mold test into something known as DRBC. That does a very good job at pushing out bacteria and growing mostly yeast and molds. Some other states, and this is because of an FDA BAM regulation, which allows you to sort of choose your own adventure on what media you grow these microbes on. But another one that's allowed in the BAM is PDA, and PDA grows half bacteria, half yeast and mold. So when you get counts off of PDA, they're always inflated with bacterial numbers. So depending on like what culture system a lab's using, you'll get 5-fold, 10-fold different answers on what's present. And then you add in these molecular tests like PCR and other tools. They measure DNA. Not all the DNA is alive or dead. Some of it is dead DNA at post radiation, and they still pick that up. So there's a lot more dispute in the industry over who's right or wrong on the total test than there are on the species-specific tests. The species-specific tests are like shooting fish in a barrel. You could add another 10 to the list, and I think you could get tests revved up really quickly, and labs wouldn't have much fight over who's right or wrong, because they're pretty easy to pin down once you nail it to a single organism.
If you had the ear of every regulator in the U.S., do you think you could clean up the problem pretty swiftly?
Well, if I had the ear, the first thing I'd ask them to do, because I'm not all-knowing on the solution here, we have an industry of a lot of talented scientists in the lab space, is put all the data public. That, I believe, will change things, because there's hundreds of people like myself or just enthusiasts who are good with manipulating data and combing through these large metric databases to find these discrepancies. So this actually is happening to some degree. The Institute of Cannabis Sciences is doing this work. They're downloading a lot of the metric data from various states that they can get access to. Sadly, most of it has to come through FOIA requests, and none of that should be FOIA. It should all be public. If it's public, then the regulators can leverage an army of people on Twitter who love to tear into this stuff and find anomalies in the field, and they'll do their work for them. I point out Yasha Khan's done some of this work here in Massachusetts and other states. He presented at CanMed this year, and he showed some really interesting data of certain labs in certain states never fail and others do, and those same labs that never fail the yeast and mold have elevated THC levels. So there's clearly some evidence in the data that's actually at the fingertips of these regulators that is only really being brought to public attention through committed scientists like themselves. So I think I would want to encourage another 100 people like them to be able to go through all of the testing data, put it all public. It should be public. It's public data. And then you will encourage an army of people to come help you find the anomalies. And that would probably be the best thing we could do for the field. I have my own ideas on which tests I think are going to paint us into a corner and which ones aren't. But I'm in a testing company, and people will throw stones at me for having a bias toward whatever a platform can detect. But I've been probably doing this longer than anybody. We got involved making these tests. I think we had them on the market in 2016. So I don't think there's anyone out there that's been testing cannabis microbes longer than we have. And we've seen all the battle scars, and we know which tests always create confusion with regulators. And it's these total tests. And if the total tests aren't taken into account correctly, you can come up with wildly different answers depending on what platform they choose. But what tends to happen is regulators get panicked and they start banning platforms, and they don't necessarily ban them on the right motivation. They just say, okay, one platform has the highest fail rate. That one must be right. Ban the ones that have lower fail rates. That's not always the right answer. Sometimes the growth media, as I mentioned before, isn't specific enough, and it picks up bacteria instead of yeast and mold, and they're overcalling. And so many of the plating systems can have false positives and false negatives. And that tends to be their default gold standard because that's what's used in the food industry. But, we're not dealing with food here. We're dealing with inhalable products. And the food industry is, it goes back to that sort of incumbent narrative presenting thing. They always present culture as this gold standard. And now that we have genomics, we can see how blind it is to so many different pathogens. It's not very specific. It's often calling things that, on yeast and mold tests that are in fact bacteria. So the gold standard isn't a gold standard for those tests. The total tests do not actually have gold standards. And I think that is the problem, is they tend to race in and ban the newer technologies that are probably doing a more accurate job, but they just don't want to face the fact that the gold, what they've relied upon in the past is now being challenged.
So would it not be on the regulator side? Could it be all the testing companies getting together, figuring out the gold standard and taking it upon themselves, or is that just not realistic? And
it's possible for them to get together and share this data, and some of that is happening. A lot of the testing labs are sharing the metric data right now to see which labs are chronically underreporting. And I don't know that will change things unless there are. I mean, there's some cases in California where I think labs are suing other labs for this type of cheating, and we'll see how that pans out. I don't know if that will persist. I think one of the challenges the testing industry faces is that they are not the large industry in this industry, if you will. It's the people who hold the purse strings and the tax dollars are really the growers. And the growers, I think, have more lobbying power with the regulators to alter the regulations than any given testing lab. So the testing labs can scream bloody murder till the cows come home, but I don't think it's going to alter the regulations, given they don't have enough of a pooled lobbying size, if you will, in terms of cash, as you do from the growers. The growers have very large balance sheets and can't afford to lobby to a degree that the labs never will. So I don't have a perfect answer for it, which is why I tend to lean in this transparency direction, because I think the more transparent the data gets, then it enables the patients to actually call regulators and say, look, we care about this, and we want there to be transparency in the data that's being produced by these labs, and we want to be able to hold certain growers and certain labs accountable for the fact that they're always fivefold lower in fail rate than the other labs. There's got to be something unique going on there, and that I think would also help the regulators, because the regulators are underfunded as well here. There's a lot of tax money coming in, but it's not all going to the regulators. It often is going to other social programs. And so the regulators have to try to navigate this industry, and they're also facing lawsuits themselves, oftentimes from growers and labs, and they don't have infinite resources. But there is a large community of people out there that want this problem addressed. Particularly the medical patients want to have tested material, otherwise they'll just go to the black market. And so they I think they're a powerful voice right now that's not heard, because they don't have access to data that can help give them a picture of what's going on.
Are you encouraged by the current state of the cannabis industry, or, are you a little bit more pessimistic about it?
I'm actually encouraged by a lot of people view the state-to-state heterogeneity in the law as being a negative. I actually view it as a positive. That I'd be really frightened if the FDA stepped in right now and declared one rule to rule the whole industry, because it would probably be the wrong rule. And it'd probably be the more you centralize that type of regulation, then there's only one person to buy off as opposed to 50 different regulators to buy off, right? So the regulatory capture thing is greatly, I think, attenuated by having a decentralized set of rules and regulations. It's painful for us as a producer because we have to make kits that are compliant with 50 different opinions on what this should be. Right now it's not 50, but whatever, it's 30 some odd states. But we learn from that. We learn from right now Colorado is doing this new thing where if you test so many times, you don't have to test anymore. And then they're going around and doing secret shopper programs and finding there's a much higher failure rate. That might be because they lowered the testing frequency too low. And I think when they lower the testing frequency that low, many labs shut down. So there isn't a strong lab economy in Colorado as a result of this. California did something different a few years ago where they did more species-specific testing for Aspergillus and E. coli and Salmonella. And that certainly lowered the fail rates. There have been some complaints that, people can see some visible mold that's not getting picked up on some flour, and that perhaps that program needs to add some additional tests to pick up the stuff that people can readily see. Here in Massachusetts, we have a mix. We've got some of these total tests in place and some of the species-specific tests in place. So we're learning from every state. And I think that's what we need to do right now in this stage, is we need to have many experiments go on and let this laboratory of democracy teach us state to state what are the right rules we can cherry pick from each before we try and set standards for the whole industry. So I don't think we know enough right now to say this is the perfect solution for all jurisdictions. I think that's a little bit, that's what people want because it simplifies life, but I don't think it's necessarily going to hone in on the most accurate answer. It might give you precision because everyone will get the same answer following the same rules, but it may not be the most accurate answer.
Are all of these unknowns what draws you to cannabis, that it's still kind of at ground zero in learning about everything about the plants and about the industry?
Absolutely. I mean, every month a new paper comes out showing some new cannabinoid they found or some different microbe that helps yield. It's a very early time to be in the field. I think at CanMed we saw one of the first cases of someone knocking out the CBD synthase gene using like CRISPR, which is an interesting case because there's a lot of GMO concern out there. But here's a case where you're not actually putting in foreign DNA, you're removing a gene from the plant, which people. You can breed to this solution as well without having to use CRISPR. This person was just demonstrating this out of Wisconsin, that the CRISPR-Cas9 system could work in cannabis. And they're able to take out the CBD gene. And with that meant there was no 20-to-1 THC contamination in the plant, which is entirely expected if you were to grow a type 4 plant. That's what you'd see, a CBG plant. But it was an important proof of principle that they got the transformation efficiency to the point where they had enough yield where they could pick out the modified plant and sequence verify it. We actually sequenced that here. I didn't even know it until after that conference. I went and looked for the strain in Cannapedia, and it was there. And you can see this beautiful, clean knockout that they performed. So that's opening a whole new window to cannabis as to what can we modify. What can we take out that is, maybe considered not as much of a GMO as putting in like a jellyfish gene, which freaks people out, right? Removing parts of the plant that may have undesirable effects is something that people may be more accepting of. So that I can see that technology working for pathogen response as well. Finding areas of the plant that make you more susceptible. Can you knock them out? And does that mean the plant is actually more resilient to these things? So it's at a very, very early stage, and the science is moving incredibly fast, which means there's never a dull day in the marketplace. And I do think it's exciting from a medical freedom standpoint, in that if we can get these regulations right in the cannabis field, we can perhaps learn from that as we move to other nutraceuticals. There's a whole host of other plants out there and fungi out there that people want to utilize in this way. There's all this exciting work on psilocybe that's going on for Psilocybe cubensis. We're seeing people look at kava kava genetics. We're seeing people look at kratom. There's just 25% of the pharmaceutical products out in the marketplace came from Mother Nature. Right, so it's a large treasure trove of molecules that are untapped, and we frankly need a mechanism to get people medicine that doesn't have this $800 million fee of going through the FDA. This is not going to it's not going to survive long. We can't keep running medicine this way. It's too centralized. So I see cannabis as a really strong, like, decentralizing movement where we can get people aware of their own health and how to grow their own medicine. And if we do it right in cannabis, we can learn from that and spread it into every other nutraceutical out there.
You've seen a lot of change in this industry over the last 13 or so years that you've been in it. 10 to 15 years from now, in your opinion, what does cannabis look like?
Oh, that's a good question. It's hard to do forecasts on politics because so much of cannabis's future is probably going to come down to some of these election cycles. But I would imagine that far out, it will probably be medically legal in all states, with the exception of maybe one or two, very puritanical states, and many international jurisdictions as well. And the recreational side of it will hopefully will calm down. I think right now there's a lot of recreational concern that there are loopholes in the 2018 Farm Bill that allow, Delta 8 and allow THC acetate and maybe even THCP and these other things getting sprayed into pens and sold into gas stations. I mean, that's an area that I think is going to backfire because it's going to get to kids, and parents are going to recoil against this legalization movement, seeing that it brought them THCP into a gas station, right? So I do think those need to. I spend my time, if I talk to kids, anyone who's interested in this, I said just stay away from anything that's not dispensary tested because you can't know what's in it. And if you're finding it in a gas station, likely it's been sprayed with something they're claiming it's a hemp pen that has Delta 8 in it. I've seen examples of Rice Krispie treats that have hundreds to 600 milligrams of Delta 8. It's just an absurd amount of THC that's it's going to create a train wreck for somebody's life, and that's not going to help our movement. So I hope a lot of that gets cleaned up by then, and we have a better understanding of what can come through the hemp industry that isn't cannibalistic to the people who are paying all these fees and fines and licenses to grow THC indoors. Hopefully we'll be able to grow some more of this outdoors. I think that's also an area that is a bit paranoid right now. We have extreme electrical costs for growing this indoors, and that means that the fees and the cameras and all the security around growing THC indoors seems to be just an added expense compared to any other crop, and it's not necessarily an environmental solution to the problem. So I hope in 10 to 15 years we can see more outdoor growing and less of this gas station nonsense, and testing costs that come down to the point where there's no longer an economic motivation to move black market material to the market.
Well, Kevin, I feel like I could talk to you for another 2-3 hours. You open up a whole new Pandora's box when we're talking about those products available at gas stations. So, I really hope we have a chance to get a chance to do it again soon. But I mean, before we get out of here, anything else that you want to make sure the Cannabis Equipment News audience knows about yourself or your work at Medicinal Genomics?
Well, certainly let's talk before 15 years.
Yes, agreed.
Maybe 15 months would be better. But so we do a lot of work on cannabis education here. We run the CanMed conference, and that's been very helpful because it gets together the physicians who have certain concerns. It gets together the patients, the nurses, the growers, and the laboratory scientists all in one conference to hash these things out. It's hard to get this done at any other conference. If you don't have all of those different bodies in place, it's difficult to get regulators to go. We're starting to see regulators show up at CanMed, which has been very helpful because we can get their insight as to what they're worried about from a government policy standpoint. But getting all those minds together helps to sort out the conflicting differences, if you will. Physicians don't want to lose their license recommending something that is untested. And they're putting their neck out already as it is, trying to help these patients with cannabinoids. So they have certain attributes they want to see. Patients tend to be very much aligned with the physicians. The testing labs are somewhat neutral in that they just want to test and want accurate rules so that they're not accused of cheating, and that they're going to be competing on a level playing field without people that are lab shopping. And the grows, I think, want the same thing. They're just often taxed into a corner and pushed to find yield wherever they can. And if there isn't a whole lot of policing on how to do this, they just go with the labs that give them the best numbers. And I don't blame them either for this, because I think they're reacting to economic forces that where there isn't real clear scrutiny or regulations on how to get this done. So getting them all together, I think, gets everyone aligned. So that's a great meeting to go to. Next year is going to be in Puerto Rico, which is being an interesting visit in and of itself, because Puerto Rico has now converted a lot of their factory warehouses down there into grows after the hurricane. So I think there's over 30 grows down there now. So it's that's a good place to see what we're doing. All of those videos we put online public for free. All right. So if you can't make it to the conference, the videos will come out eventually. They're usually a couple months delayed, as we have to get them the PowerPoints kind of merged in with the video content. So there's some editing involved, but you can see the last 6 years are online if you want to page through those. There's great presentations from physicians and chemists and lab staff and growers. So that's one area to follow us. Medicinal genomics, you can find me at. We have there's oftentimes I write about hoplite and virus stuff on my Substack and on a Twitter account. But we also have a database here called Canopedia that has about 2,000 genomes public. And some of those now have their microbiomes published. So we've got about 1,000 genomes that have been sequenced whole genome style, where we have the microbiome background. So we know, with probably better than anyone in the world, what are the prevalence of different microbes on the cannabis stems, flowers, leaves, and roots. We've sequenced all those tissue types. And that can help inform regulators on, all right, what are the real pathogenic risks here? Maybe we just make tests that target those and forget about everything else. I mean, right now they're talking about putting listeria into the works, and I've never found listeria on a cannabis plant. Maybe it gets introduced through, cooking practices in kitchens and milk and whatnot, but it'd be the lowest on my list of things to worry about in cannabis. Yet it's probably going to be the next thing that comes in, because the food industry likes to just copy what they've done before and not have to change anything. I think as a cannabis industry, we can do better than that. So check us out there. Medicinal Genomics and CanMed are probably the two best places to follow our work.
When is the next CanMed, and what would be the highest on your list of priorities?
So on other pathogens, I would say so many years ago, we published penicillium. There's a couple penicillium that are found very frequently that make mycotoxins, and no one's looking for their mycotoxins. So Penicillium paxilli and citrinum. They make Paxilin and they make citrinum. And those two mycotoxins, one of them has some published contraindications with CBD, and the other one can be seizure forming. So those are found very, very frequently on cannabis, and no one's looking for them. And the mycotoxins aren't being screened either. And the other one would probably be Fusarium, because Fusarium, it's bad for the plant, but there have been cases of fusariosis. It makes some nasty mycotoxins and can infect open wounds. So that's frequently found in cannabis. And no one's really, the growers do look for it because they don't like it affecting their yield, but it's not anything that gets looked at in a final product standpoint. And it is something we think is playing a role in hoplite and viral transmission. So I think that's an important one for the field to know about, much more so than listeria. I don't think we're ever going to have a really high positivity rate for listeria, unless it's in like an infused product. I don't see it showing up in the plant. So that database can very easily help inform regulators on where the threats are, as opposed to just lifting the threats over from a previous industry.
And what month is CanMed is? CanMed
CanMed is in this year. It was in May, but I think it's going to be pushed the first week of June next year. Okay.
That sounds like an incredible opportunity. I hope I can make it, and I hope many others can make it out to Puerto Rico next year.
Excellent. Yeah. Well, thank you for the airtime here. And if you guys have any questions, hit me up directly, and I'll send you the link for that preprint.
All right. Well, before we get out of here, please make sure to like, share, and subscribe to the podcast. You can help us out a lot by leaving the podcast a positive review on whatever platform you use. If you want to email the podcast, you can reach me at david@cannabisequipmentnews.com with email the podcast in the subject line. You can also subscribe to our daily newsletter. Make sure you get it delivered to your inbox first. For Kevin McKernan, CSO and founder of Medicinal Genomics, I'm David Mantey. This is the Cannabis Equipment News Podcast, and we will see you next week.
Welcome to the Cannabis Equipment News Podcast.